I prepared this presentation today a bit different. This is why I call it the backstage story of a project. I thought it would be more interesting if I would show you more like the step-by-step of which decisions I took, the bumps on the road, and how I solved some of the issues. And I hope you like it. Let's get started. So first of all, you might be asking yourself why the only Brazilian person in the group is doing a specific project on the Czech flora. And why am I doing this if last semester I presented you a completely different project about communities varying through time? And this is exactly the reason, because I had my main project, which I had to go to the field every month and collect my data. So meanwhile, I wouldn't have anything to do. So I couldn't be just going to the field once a month and that's all. So for this reason, Milan introduced me Palladia's database. If I had any idea or what I could think about a secondary project or a parallel project to study, meanwhile, and this is how I got here. This is how I end up working with the Palladia's database and the Czech flora. So the main data, or the data I used, was from this grid, the grid in the Czech map, in which each one of these cells in the map we have presence absence of species. So around 2,700 species, 2,300 cells. What else we had? We also had characteristics for the species, a list of characteristics including functional traits and other ecological aspects. For example, where the species come from or which type of habitat it lives in. And this is when I realized, okay, they have functional traits, they have the species list, and functional traits are a universal language to work with vegetation. Not only for me being an outsider, that I don't know many of the species, but also if we want to communicate with people working in different ecosystems and also to relate with the environment, sometimes it makes more sense to understand which of the characteristics of the organisms are better suitable for that habitat than knowing exactly of the species. So yeah, there are many papers and very nice studies around where people are taking these huge databases and mixing data of functional traits and get very interesting trends and very interesting discoveries. So I could think something that I could do. And this is this was a bit strange for me, because most of my time I was used to have a question or a hypothesis and then look for the data to answer my questions. But this time I had the other way around. I had the data and I had to find a question to study on the data that I already had. So this is when I got presented to this paper. Was still being written at the time. Now it's accepted. And in this paper, it's only like showing all the nice data we have in the Palladia's database. And in this paper also, there are many of these maps, colorful maps, showing for each one of the traits the means of the traits in the cells. So we have both for binary traits, for example, here annual herbs. So this is the proportion of annual herbs in the map. But we also have for quantitative traits, like the mean length of the flowering period. So the color represents the mean of all species in that square. But the thing with this paper that I was also invited and I participated in is that it has many and many of these maps. Many, you tell. I didn't count, but I think it's hundreds. And you can imagine how difficult it was to kind of get into an understanding, a holistic understanding of what was going on and how can I put together all of those traits. So how can I summarize all this information in a straightforward way? I already worked with functional traits, and I know that working with multiple traits can have some issues. The first issue is the one that I already told you. If you are working with multiple traits, it's very hard to have a whole understanding of what's happening. In a data set with multiple traits, it's very common also for you to have absent data, either because the data wasn't collected or is not available, or sometimes just because the plant doesn't have that trait. For example, seed traits in plants that doesn't produce seeds. The other thing is that the traits are also correlated. They are not independent from each other. The traits usually represent some strategies. So there are impossible combinations. That means that there are trade-offs or choices in strategy the plant have to do. But exactly because of this reason, this is also a solution to my problem. This was interesting study where the authors managed to put thousands and thousands of species described by only 6 traits, and they got most of the explanation on the variability of the traits here. So we can see the strategy, we can see the trade-offs, for example, from height and small plants, and also the leaf size. And it's this is not new in functional ecology. Actually, since the beginning, I would say people would do this, would do an ordination, but instead of using the axis for the analysis, they would rather separate them in functional groups and use the functional groups instead. Even me, I already used this type of method using the axis instead of the traits for some analysis. For example, here, how these strategies are answering to a disturbance gradient. In another paper that I published, I have how the community-weighted means of those traits are affecting ecosystem processes. For example, here it's the change in biomass. So yeah, now I have the first aim of my project. I want to define the main axis of variation in plant traits as surrogates for functional strategies. So I'm going to build a functional traits ordination, but first I have to select and prepare the data. We first had to select which species are going to enter in our analysis. So we selected all of them from species level, except for these 2 genera, where we choose the sections. We also excluded some species because they were extinct, or they were casual aliens, or only cultivated. So this is the final count of species. And also for the traits, as I told you, we had not only functional traits in the dataset, we also had those other characteristics which are not considered functional, or we couldn't see ecological meaning from them. We also added 4 traits which are very important and was not included in the Palladius database in the beginning. Leaf size, specific leaf area, leaf dry matter content, and seed mass. We also had to separate the categorical variables, ranging from 0 to 1, for example, in different columns. This is called the fuzzy coding because some species could have intermediate values. And for some of the categorical traits, we also merged them for not having so much columns. So this is an example. Many pollination syndromes that are aldogamy, we just merged them into autogamy. So we didn't have all of these columns. So in the final table, we had 69 numerical traits or individual binary variables. That is that expanded variables that I explained before. Okay, and for calculating the ordination, the principal component analysis, we also had to transform some of the data. For example, we had to log transform some qualitative variables. For the fuzzy coded variables, we had to standardize by the total. So we wouldn't have different weights for different species because of that. And also for this binary, we had to centralize and normalize. So we would have an equal weight for each of the traits in the fuzzy coded variables. For example, growth form. Growth form is one trait that was separated in multiple traits. So we didn't want each one of these columns to have the same weight as a trait that only contains one row as, let me see, carnivory. So we would decrease the importance of each one of these fuzzy coded variables. And also for the missing values, we would replace for the overall mean of that trait. That means that NA wouldn't change the final calculation or the final importance of that trait in the ordination. We used vegan package, the function RDA in the R software to calculate the ordination. So this is our first result, which shows the first 2 axes of the ordination. Here we have the most related traits to each one of the axes. And here we have the species that are also more related, or a subset of the species showing all the range of the trade-offs. And here I wanted to highlight first, in the first axis, it's separating mostly the annual herbs, which are not clonal, with clonal herbs. So here we have plenty of clonal traits, for example, clonal index, clonal multiplication, even vegetative reproduction. So this is the main axis, the main differences in the first axis. And in the second axis, we have the separation between plants by their size. So here we have height, we also have shrubs and trees, macrophanerophytes, and here we have the smaller plants. Here also in the second axis we have the leaf economic spectrum, having SLA in the positive value and LDMC. The leaf economic spectrum represents an axis of investment strategy of the leaves. In here, the plants would be, let's say, more conservative, and here they would spend more energy and produce more leaves that are thin, that are not so strong. And also we could finally produce, similarly to the maps I showed before, we produced maps of the scores of the species. So instead of having means of traits, we have means of scores. So for example, here is the first axis. So the most red, the square, the most it represents the positive values for axis 1. Which are here the clonal traits. So the red ones are more clonal, and this is more annual species. And the same for the axis 2. We have the positive values, which are the small in size. And the plants that are higher in the blue zones. So you as a native from here probably have a better understanding than me what can be the behind these trends. But I also wanted to check which environmental variables are leading those trends. I was given a set of environmental variables that I could use to analyze what's leading these changes in the functional traits. So now I have my second aim. I wanted to explore how the functional trends vary with environmental factors at the country scale. And here's where I got, I would say, my first bump in the road, which is that actually the historical and evolutive factors, they affect the functional traits. And so there are still discussions whether you have to exclude this importance or you can just ignore them. I decided that I wanted to incorporate in my analysis because I didn't wanted to have mix these historical factors with what I was seeing in the trends. So how can I account for the phylogeny? The method I chose, because we were dealing with an ordination, was basically we had to first build our tree for the species that we have. So we use this phylomaker to produce a tree with distances between the species. With this pairwise distance, we build an ordination only with the phylogenetic distances. Here we are not accounting for the traits anymore. And then here I did this analysis, the broken stick, to check which one of the phylogenetic axes were meaningful. So it was around 50 axes. So we used all of these 50 axes as covariates in the PCA. So we basically run the same PCA as before, but including this axis in the PCA. So here is the result again. It's basically the same thing. The phylogenetic axis didn't affect so much the main trends. It changed a bit which traits will be more important here, but the same would be in that region. Again, the axis 1 separating more annual herbs from all of those clonal traits, and axis 2 separating big plants, trees, height from the small plants. Also SLA is here, LDMC is still here. One interesting thing is that we have kind of a grouping here of annual herbs, green leaves in the winter, flowering length, all together. And we also decided to check or to explore more axes this time. So I included 2 more axes, axis 3 and 4. The axis 3 represents separation in the negative values of, we can see here, there are rosettes, myrmecophori, hemiparasitic plants. And here we have parasitic epiphytes, fleshy fruit, woody liana. And the PC4, we have a separation between the parasitic plants again and geophytes. In the negative values, we have dwarf shrub, plants with green leaves persistent, and simple leaves. These 2 axes are a bit more tricky to try to interpret, but I believe we can disentangle this a bit more in the further discussions. So, but what I was important by the moment was how could I build these models? I already had my response variables, which are the means. Again, we calculated the mean of the scores of the species for each one of the cells. So we have 5 response variables. Here I also included a measurement of functional diversity, that's raw entropy. So the raw is calculated for the 4 axes, and the higher the number is higher the functional diversity. And here it is the maps again. As I told you, for the first 2 axes, accounting for the phylogeny didn't change much. And here we have axis 3 and axis 4. And here is the functional diversity raw index, showing that in these regions we have smaller diversities, and this central area or these river basins we would have higher diversity. And as I told you, we had available quite a big set of environmental variables to test, which I didn't know exactly where to start, so I wanted to start with all of them. Here are the ones that I think it's usual: altitude, temperature, precipitation, terrain ruggedness, and topographic wetness are topographic measurements. For example, ruggedness will be how rough is the terrain, and topographic wetness is the concavity of the environment, so it means that it can retain more water. We also have this difference from precipitation and evaporation. That means a measurement of drought. we have continentality, and we also have the percentage of the cell that's covered by forest, arable land, water bodies, and we also have the percentage of the cell that's covered by different types of bedrock. So here are the types of bedrock. We also wanted to test how they would affect the functional traits. So first I run a stepwise model selection for each one of the response variables separately. I use the step function in R, and from the beginning, the 19 predictor, for each one of them I had 10 to 12 selected, which is quite a lot. I think I don't have to say that the issues concerning models with that big amount of variables selected. And if I did the linear regressions, most of them will have the significant P. So here's my second bump on the road. Is that easy? Are those relationships real? So this is where I want to show you these maps. I believe that knowing the Czech Republic, you can try to understand the trends here in those traits. And there are trends, like you can see the differences in different parts of the country. But I have to say that these 4 maps are made with random traits. So this was an issue that David Zeleny presented to me when he was visiting our group in 2019, I think. And I got a bit scared because, wow, you know, we work with community-weighted means, and somehow now it's shown by your eyes that your trends can be just given by the co-occurrences of species. So there is this one paper that shows, like here in North America, all of these nice trends are also caused by random traits, as I understood by the paper. They use birds and plants. And this issue was also described in this paper. So are we having too optimistic results from the community-weighted means? So first we have to understand how community-weighted means are calculated. First you have, as I told you, the species described by traits, and then you have your community described by species, and then you multiply one matrix to another, and then you have the communities here by the traits. So this is the community-weighted means matrix. And when you want to relate these things, you only compare these 2 matrices. Like you completely ignore everything that happened before, how this was calculated. So if you want to test traits, the traits were inputted here. So this matrix was generated. So this is how you ignore everything, and how you usually test this relationship. As I told you, you usually ignore what was happening before. And for example, if you are going to do a permutation test, you are going to shuffle one of these columns to generate a P. However, you cannot ignore the traits that was in this matrix. So a better option would be to generate a p-value based on shuffling your trait matrix. So in every iteration you are going to shuffle your trait matrix, multiply with community data, and calculate community-weighted means every time. So this is what I did. I had a help with a former colleague from Brazil, and he wrote a script that is based in these two R packages, where he generates this shuffle in the trait matrix and calculate community weighted means. For you can say how many times. I did it 999 times to generate the P with shuffling the trait, and not the, let's say, the normal way, or how you will do. Like here, if you enter R, or if you enter your program, you usually enter with these two matrices. So for testing this, I had to enter with this and this matrix. This one was calculated in every iteration. So this is an interesting result that we have. So here I tested separately each one of the response variables for each one of the, each one of the predictors for each one of the response variables. And we can see that some of the relationships that done by the normal way, or the way we usually do, they were significant. But if we do the trait shuffle, it shows that it's actually not. So for the first axis, actually just two of them didn't have the significant P. But for the other axis, for example, axis 2, we excluded most of the variables from the model. And for PCA 3, the same. So we ended up with only 3 predictors. And for PCA 4, also we only ended with 3 predictors. For RAW, I showed the whole example with community weighted means, but for RAW is the same idea. You can do it the same way. And we also had 3 of the variables that was, here I did only 99 shuffling, but anyway, you can see that the P's we got in the beginning was not real. They were just because of those species co-occurrences was causing it. So I thought I was done. I thought I had my models. I thought, okay, is that easy? But then I realized that I'm working in a geographic scale, and all of my plots are like neighboring each other. They are contiguous to each other, and it's probably wrong to just analyze it as if they were independent from each other. So I also had to include the spatial autocorrelation, which I use these DB means, which is calculated distance between the cells. In our case, they are contiguous, and the distance between the non-neighboring, or the most far away, is given a higher value. So with this distance matrix, we calculate an ordination. So this ordination will have different axes, each one representing a different spatial scale. For example, the first axis usually will show the differences in a smaller scale, for example, the neighboring cells, and some of the other axes would represent bigger distances. So again, we did a selection of axes. For each one of the response variables, we wanted to know which spatial axes were leading the trends. So we run also in R, and we use the package add the spatial. So again, for the spatial, for accounting in spatial variability, we run a variation partitioning, where we wanted to know how much of the variation was given by the spatial component. Here we use the spatial axis that we selected before, and here we would enter with all environmental variables that were selected for this or that response. So as a result, we had the environment alone explaining only 4 to 9%, the space alone representing more, like 11 to 37%, and the environment within the space or the covariation between them was represented a big part of the explanation, 16 to 52%. So here I prepared small presentation of each one of the axes, what's going on, to understand a bit better. So here is for the axis 1, where we have the map. Let's remember that the green, the red areas are more like clonal traits, lateral spread, while the blue ones are more like annual species, herbs, pterophytes. This is the variation partitioning. So for this one, we have like most of the explanation is the shared part. This is the R square of the variation partitioning. And here I have shown each one of the environmental variables, how they are affecting this axis. So here, for example, temperature. We have in the lower temperature more related with high values of clonal index and lateral spread, while we have in the smaller temperatures, sorry, more annual herbs and pterophytes. Topographic wetness is also negatively related, and these variables are positively related. For example, oh, sorry, this one disappeared, but terrain ruggedness is positively related. With here would be the clonal index, and here would be the annual herbs and pterophytes. The percentage of cover in forest in the cell. And this is the shuffled P for the whole model. So including all of these variables that I told you, this is the shuffled P. For PCA2, we also have a great explanation from the spatial component and the shared component. And we also have it's responding to the topographic wetness, the percentage of water bodies, and negatively related to this axis, we have the bedrock, tertiary volcanic rocks, and the terrain ruggedness. For PCA3, then we have also the water bodies and the topographic wetness. And the PCA4, we have, again, the topographic wetness, water bodies, and forest. And for RAW, the trend is usually like this. You have a small share of only environment, a big share shared, and the spatial also explaining quite a lot. RAW had many significant, the model containing RAW as response has many significant environmental variables. So it's responding to most of them. Here it's responding negatively to them, and this is the P for the whole model. So the conclusions for now is that the use of ordination axes as surrogates for species strategies simplifies the trait information, though I think I still can dig on what exactly each one of the strategies are representing. The use of community-weighted means and routes shows significant trends in response to environmental variables in broad geographical scale. However, I think I still should dig further into these models. The first thing is how to combine the model selection with the trait shuffle P, because the model selection selected those variables that are actually just biased by these co-occurrences. And here I got into a circular problem, because which one of the tests should I do before? Because should I test all of them by trait shuffling beforehand and then do the model selection, or doing the model selection first and then testing was the best option? I think still there is a place for discussion in this issue. And another thing that just I realized that are all environmental variables propitious to such analysis. I realized that probably I had to do a pre-selection of those variables before. For example, here, percentage of volcanic rocks. Like all of my points are here. Like, is it? Can I trust this relationship? Also here, and the water body is like maybe some transformation. Maybe I shouldn't select this environmental variable at all. Another thing that just came to me when I was preparing the presentation is that I have 3 environmental variables that are not independent. For example, this P divided by PET could be represented by intersection between precipitation and evapotranspiration. And also, are all axes meaningful? So I decided for this second set of analysis to include axes 3 and 4, the functional axes, but it's quite hard to interpret them. So what do you think? I think there's still place for discussion. I wanted to have feedback. I understand that I did a lot, but there is still quite few steps to take until the final shape of the project. Thank you for your attention.
Okay, so thank you, Felícia. You did really a big amount of work. I can see, and it can be really difficult to, I don't get it, and to dig into the big data set. So
yeah, I was never used to do this. It was the first time I had to deal with this huge data set.
Yeah. So it's time for discussion. So Milan, you was the first one.
Yes, thank you, Felícia, for the presentation, and especially I see you really made a big progress. So thank you, and I think you are on the right track. I have a couple of questions. Like first, when we were preparing the ordinations for the Pladias report paper, some authors commented that the results of the ordination and why it is different from, for example, what Diaz and colleagues got in their ordination is that we simply have too many clonal traits. So if we put too many clonal traits in the model, then there is a higher probability that these traits will appear as the most important, and actually they did, because they drive the first axis. So what can we do about it? Or
so I also thought that I know that statistically is an issue to have so many correlated traits and traits that we know that they come from the same data. But I thought that doing an ordination, we would have at the same time all of them together, but we could see how this group is separating from the others. So I don't think it's an issue in the sense that it's obvious that you have them all together, but it's to place all of them in relationship to the others. If it's an issue, we can try to decrease the weight of them the same way we did for the dummy variables. We can decrease the importance of them by means of transformation. I believe this can help. But,
yeah, but then it would be maybe quite arbitrary.
Maybe just dividing by the number of clonal traits that we have.
Okay. I see there are several people who want to ask questions. I have more, but I will now give space to them, and then maybe I will ask again.
So you will be comment on
just quickly. Sorry, I think that with PCA and with this kind of analysis, it's always arbitrary. There is no simple solution, because we can take it other way around, that Diaz et al. Results were impacted by the fact that they didn't have any clonal trait in the analysis. So that's why their results are worse than ours, because we have clonal traits, and that there is no. This is always arbitrary. Yeah. So, and I'm afraid that there is no solution to this. Okay. So I will probably continue with my question. Or my point, and that is that with this direct ordination analysis, or basically model between environment and traits, it may be worse to check really to leave out this step of the PCA and basically use traits as the raw trait table as a response, and the predictors as you did. So you would make an RDA in this case, and maybe that would actually simplify the thing, that you wouldn't need to interpret the PCA axis that much. So you would get more direct relationship between traits and the environment. In this case, you would need to include this phylogenetic information as basically commutative means of individual phylogenetic axes, but it's possible. So, well, I don't know. Maybe the result would be just chaotic, and maybe also impacted by the correlation between the traits, of course. But it may be something which could be worth checking.
Okay, I think we can discuss this. Yeah, I don't know what to say about it.
Okay, so hi Lars. It's your turn now.
You are mute.
Yeah, your microphone.
Now I should be unmute, is that correct?
Yes.
All right, perfect. Yes, maybe to, for now, stick with the way that you did the analysis. I mean, yeah, I also have ton of questions. But so let me maybe pick one or two, which are also more on the technical aspects probably, but one maybe that related to the things that you said in the very end, already when looking at these graphs of the single environmental predictors and the PCA axis of the community weighted mean, yeah, I think it became evident that there may be some transformation of the variables should probably happen. Anyway, you have this kind of thing, right? These are percentages that are covered by certain whatever geological type or usage type in percentage. So you might need some special transformation there. So square root might do the job to make this.
The thing is that I was so much. I gave so much time to the shuffled P issue and the spatial issue that I forgot the most basic thing is to check my environmental variables before. Like
you lost track of some other things.
I really think we should pre-select them or do the transformations that must be done.
Yes. In the same respect, I was thinking, especially when you look at the functional diversity at this scale, I think what might be useful in there, and maybe I have missed that there is some variables that already express this, but if you would have some kind of approximation there as an additional explanatory variables that kind of estimates, let's say, the amount of habitat heterogeneity, like the number of habitat types.
That makes sense.
I think that could probably be an important predictor in the particular case of RAU, because you would probably expect that the more habitat heterogeneity, the more functional diversity in your traits you would get. And then maybe to go back to the very beginning, one thing was that your PCA plots before and after putting the phylogenetic eigenvector axis basically look kind of similar. Did you check comparing these two whether the explained variation on this first couple of axes got higher once you put in the phylogenetic eigenvectors into the PCA?
I'm not sure now because I did this step so long ago. I have to check also that.
Yeah, it was just something that kind of I was curious about. Maybe to see this would be so. Given that they don't look much different, probably there's not.
If the phylogenetic axis improve the relationship between what again?
Not the relationship, just so for each of the PCA axis, right? You have an explained
increase the explanation of the axis.
Yeah.
Okay.
Single axis, exactly. Yes. Okay. I guess I stop for now because there's other people who have some questions, and then we can also.
There will be time to get back to you.
Yeah,
after.
Exactly. We communicate.
So now, Padu, it's your.
Good morning.
Hi.
Hi. Very nice talk, Felicia. Thank you. I also run into some of your problems using my data, so I know how sad can you feel sometimes, but I guess it's rewarding at the end. I also will try to be brief. Few things or comments that I wanted to ask you. The first one is that you said that you just use overall mean values for those that were missing, and I don't know. You didn't say how many species really had missing trait values, but I don't know. Like if using the mean of the genus or the mean of the family would have been more appropriate.
It's the total mean of the whole column.
That's what I mean. That.
Perhaps... it's not imputation of data. It's just we couldn't leave that cell empty. So this is just a number. The average will be a number that wouldn't affect the final product. It wouldn't affect where that species is on the ordination, and it wouldn't affect the weight of that trait or anything.
I understand. But I'm just wondering if that is the best approach then, considering that, for example, for plant height, if you know that is an herb or a grass, and the mean trait value is like I don't know, like 1.5 meters, it's not real. It would be more real to use a more appropriate trait value for that species. That's what I mean. I'm not saying it's better. I'm just saying that I don't know how the results would change if you would use a different approach.
Yeah, so we decided this over the imputation based on phylogeny because we use the phylogeny to extract the importance. So we couldn't put phylogeny in the both ends.
Okay.
So this was actually just a way because in this RDA function we cannot enter NA data. I discovered that there are ways in which you can calculate PCA with absent data, but I didn't know in the beginning. So this was just a way for not having that cell empty, and we wanted to have the minimal impact on the result. And this is the way we found, like, in theory it should work, but I have nothing to compare to. Well, maybe I can compare to the same PCA considering the NAs.
Okay. Then something more conceptual, like when I saw this list of 69 traits, it was like crazy. And for me, it's very difficult to really make try to imagine like how this axis would like, even if you like get very clear like explanations of what's going on the axis. But I just think that if you have like such a big list of traits and you have no previous hypotheses on how environment is going to affect one trait or the other, then maybe you can somehow reduce that list of traits to something that is more meaningful to ecologists or botanists, and that still keeps all the variation that you want to keep in that matrix of traits. For example, I think clonality, as Jakub was saying, it's important because it's usually unexplored, but maybe other traits are not really that meaningful and are just complicating the picture that you are inputting in the PCA. So just as you were saying that maybe you have to decide what environmental variables you want to choose, also like what traits would be maybe useful. I don't know, it's just a comment.
Mm-hmm.
And one thing I wanted to say, maybe it's wrong, but you talk about community weighted means, but I think it's not community and it's not weighted.
Sorry?
So I think
they are not weighted. It's just community means.
Yeah. Well, so we could argue that they are not even communities, right?
Yeah.
So it
it's
would
just
be
because it's usually called this name, and I had the method to explain with community weighted means, but yeah, it's cell means or the means of that.
Or assemblage mean or whatever.
Yeah, I mean sample means.
And I'm just saying this because someone told me that before to me, and I was like, okay, I get it. And it was in a review, and then I had to change everything. So it's better to take.
Because the traditionally the name of this calculation is community weighted means, but doesn't mean it has to be a real community.
Good. And then another comment. I have others, but I will also keep it short. You might have it's a methodological thing. Like, have you considered you have a very large sample size? So I'm wondering to what extent these p-values you are showing are really significant or affected by the large sample size that you have, and perhaps using confidence intervals or something like this would be more meaningful in your case. And also, and related to this, you also show these bivariate plots. And I wanted to ask you if that line that you plotted is the result of the fitted, or is just the linear regression between the two variables? Because
no, the figure is just the linear.
Okay, because in that case you are like not including the effect
considering the others. Yes.
Others in the and also the spatial autocorrelation, blah 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 blah. So just to and well, just this for now. Thank you.
Okay, so thank you. I guess that because of the Platea's structure, there are not so many gaps in the data set, and I think that there are just few empty cells in the matrix, but this is just my guess, I would say, to respond. But Tomáš, welcome to our meeting. Hi, it's your place.
Hi, thank you. It's really nice to take part, and I really enjoyed your talk, Felícia, I should say. And I have a bunch of comments, which some of them I'm going to send you by email. Perhaps I would like to tackle 2 issues or 3 issues that have been already discussed, and to which I have something to add. First is the trait selection. when checking the traits that you are using, I think that there are cases where, because the Plagias arose as a essentially merger of several databases, there are traits that are essentially duplicated. Like if you look at pterophytes and annual herbs, this essentially says the same thing. Mm-hmm. So I don't think it's a point having these two as separate traits in the analysis. And this would make things, and there are many, not many, but quite a few pairs of traits. And I think this should be double checked by the people who provided these traits to make sure that we are not essentially using duplicate information. And part of this concerns also clonal traits, since also some of the clonal traits that we are using are very correlated. Perhaps we could use correlation as a means to delete some of them, or we could just use a sort of conceptual thinking, which one should be there or not. So this is one thing. I think this really should be redone. The other thing is the trait imputation. I don't know, as Zdenka pointed out, how many of sort of imputation cases were there. Perhaps Jakub would know. If you use this column wide imputation, you definitely reduce phylogenetic signal in the data. And for traits that are phylogenetically constrained, and some of them really are, as Lars, I don't know who it was, pointed out, this could make a problem. And there could be a way how to check this actually. Since one of the potential other approaches that one could be using would be to calculate phylogenetic PCA directly with optimization of the lambda value, which would actually check the phylogenetic signal in the correlated set of the data. And perhaps one could see how this lambda changes depending on the different approaches one is using in the data imputation. So that was another comment I wanted to make. And wait a minute, I just made a list here of comments. I think the other ones are not very important or have been tackled by other people. This log transformation is important thing, or a square root, both also for predictors like water bodies or something. But I think that these were the most important things I wanted to comment on.
Yes.
I will send you a couple of comments by email, but these are more technical things. Thank you.
Thank you.
Yeah, thanks, Tomáš. And yeah, Milan.
So yeah, I think. Yeah, I think these are quite important points. So, what to do with these correlations? So, don't you think that it's solved by PCA? That let's say Felícia would calculate a PCA as she has now, and then from each axis of PCA, we would select, also having in mind some, let's say, background ecological knowledge, which traits could be used for that axis.
I would go first a priori. I would a priori first think which traits are not meaningful to have them both. Like annual herbs and pterophytes, this is a clear example, but there are more of this kind. And then I would perhaps take the approach that you are proposing. But also what one could do is to calculate the overall prediction of each of the traits by the rest of the traits. And something like variance inflation factors are calculated in the regression analysis, and you use this to select the traits that perhaps should be excluded after the a priori selection.
Mm-hmm.
If I can quickly make also one more comment on this, basically bringing together these two things, the trait correlation and the imputation, you could theoretically, if these NAs are really a big issue, impute trait values from the trait structure, from the trait correlation.
Good point. Yeah, good point. Good point. Yeah, good point. One could merge two traits to make them one trait. Yeah, this is a good point.
Do you think, let's say, if we took just means for the genera, would it be really so important for like subsequent use of phylogenetic information? Because I think means for genera would work quite well, and phylogeny is not just genera. A large part of the variation in phylogeny is due to higher or deeper clades, let's say. So it would be some, let's say, a compromise way, and not like exactly correct, but maybe, well, I don't know.
But one is not doing phylogenetic reconstruction. You want to get something that is most likely to be close to the real value that is not available, and then means of genera is something that perhaps would work.
So you wouldn't mind.
I wouldn't mind. I wouldn't mind. And
I wouldn't mind either. Okay.
But this also could be tested. Just one sentence. But this could be tested. You could just compare what happens if you have a phylogenetic PCA with overall imputation or imputation by means of genera, and compare it by the different lambda values that one would get. Sorry, Jakub.
That's another point that in my experience, actually the PCA axes which are used in this phylogenetic eigenvalues analysis basically do not reflect relationships within genera, because when you do this selection of the axes using the broken stick model or whatever method, so these first 100 something basically show relationships within families, between genera within families. Let's say these higher level axes, but not relationships within genera. So yeah, this makes sense to use the means within genera. That's a good point. Yeah,
but also make a comment to that. I think that if you will sort the traits from those which has the less empty values and those which are just filled by the means, that still the most important in the PCA will be those which has the less empty values, because they are not so random as those which has a lot of empty cells. So I think even if we will fill it, and then we will choose the traits which are the most important according to PCA, we will have those which has a lot of data. I don't know if I said it somehow understandable, but this is my impression from the calculation.
Yeah, I think Zdenka, you're right. Yeah, and in general, I think that the number of NAs is quite reasonable in the data set. It's not a big issue. Therefore, we didn't made much effort to use the best method, and left this for this discussion. And concerning the trait selection, I agree. You're right that we should do some kind of a priori selection. For this page, I think it was quite important to show the correlation among all traits in the PLADES database for us, like internally, to see these correlations. And then we should decide which to use, because it's not so simple that, of course, annual herbs are the R square between them is basically 0.1, but on the other hand, these are components of life form and growth form, and we should probably decide which trait, whether life form or form we will use, and then go for that. Yeah, and well, it's quite arbitrary, and I myself don't know which one to use.
But okay, I think we can. Sorry, Icho. We can do it. So if Felícia, if you produce a matrix with correlations between traits and trait states, we already have some knowledge from the previous ordinations. So I think we can do a meaningful selection of a smaller subset of traits.
Yeah.
So maybe prepare this, and we will look at it.
Maybe it would be nice to have this matrix, and together with that, now NAs in each trait.
Yeah, Irenka is there. Yeah. Good job.
I wanted to say that the number of NAs now is about 10% in the matrix. I know it, and the most problematic seems things like seed weight or something for plants that do not have seeds, you know. So we have there this Dryopteris, all these ferns and all the other who do not have seeds. So maybe we should also think about how to use the data there, which can't be easily computed. And another point, Felicia mentioned that there are 69 traits, but basically there is only half of the traits. There is around 30.
69 columns that we use.
69 columns
because were expanded.
Yeah, many of them are just coded as dummy variables or fuzzy coded variables. So
in the ordination, they are one separate thing.
Yeah, and I had another point when you included these bedrock types and you use them as separate predictors. Wouldn't it worth like using it also as dummy coded variables or fuzzy coded variables, where it would be I don't know, the prevalence in the grid of the bedrock or something like that, that it wouldn't be that like analyzed separately, but as one predictor bedrock. Just a suggestion.
Yeah, because we have different types of bedrock and different covers of that type of bedrock within the same cell. So I don't know exactly how we could put those two informations together. Maybe we could just use as a dummy variable the bedrock with most cover in the cell. I don't know.
Okay, so Milan, maybe we can move to other questions.
From what I said, I understand that ferns now have seed mass as the mean of seed plants.
Yes.
Okay, so I think it would be better to replace the seed mass for ferns by the minimum seed weight for seed plants. Yeah, so that's one suggestion.
Why not to use spore weight directly?
Well, we don't have them.
We don't have them.
Well, sorry, can I just add something? Well, we were discussing this quite thoroughly, and actually we use zeros for this cases initially, but and we actually decided to go for this way because it's well, technically, using this mean shouldn't impact the correlation structure that much. But if we use zeros or something like that, then we would just get the distinction between ferns and the rest of the species. So it would be. Then the seed size would be quite uninformative, yes, because we would get just this contrast, which would override everything else. So it's quite I don't know. There is no ideal solution for this case, and there are more cases like.
I remember that the discussion we had back then. It was more like. Okay, we are not so much interested where these or that species are gonna be,
exactly.
but more how the overall trend of the trait. So we thought it would be a minor noise, or like it's not a so fine resolution. It's not a thin resolution which we are working. We are working with bigger trends. So these details. wouldn't be, the weight of them wouldn't be so big. I don't know. I remember we having this discussion in the past.
Okay, so Irenka, now Padu, and then me, and then Irenka. Yeah?
Yes, I just wanted to say that we just prepared a study or a paper at the European scale where we had the same problem with this seed mass issue for ferns. We ended up using as Milan was saying, the smallest seed mass in the whole data set for ferns, but we also tried the spore mass, which we took from a paper that we found somewhere about spore mass in ferns, and the results didn't change. But of course, the number of species was different than in your case. But anyway, it didn't make much difference. What I wanted to say is that, like, for someone like me who is not really aware of the vegetation in the Czech Republic, maybe just like you, I was thinking that when you want to publish this, I would like to see these maps, at least the ones of the axis, next to a map of the habitats of the Czech Republic or the vegetation in the Czech Republic, because actually with this axes are, in some cases, what these axes are telling you is, in these areas you have forests, because the second axis is mainly trees with very conservative strategies, and if you put that next to a map of the habitats of vegetation types in the Czech Republic, you can get a real quick picture of what's going on there. So I don't know if that would be useful, or you could.
Okay, yes, totally. Because as I told, we are working in such a big scale that we are getting this difference from forest to non-forest. We are not getting the thin difference between like different types of forests, for example. Yeah, but it would be interesting to have at least an idea of the vegetation.
Yeah, so my question is also going to the final version of the results, because you will have a lot of maps, and then you showed us some linear regressions, and it will be maybe better to have something much more complex. So something like regression trees, is it possible to do it that the first division of the cells is based on annual species and perennial, and then something like that? So maybe if it is possible, or
yeah, I will have to think that.
Yeah, Iranka, Milan. Yeah.
I just wanted to get back to the point with the ferns. Maybe we can produce also some PCA without pteridophytes, just to check the patterns. There are not that many.
Yeah, Milan, maybe. Yeah.
Or simply we can take this database that Padu used for the paper and link it to Pladias and use it in Pladias and in this data set. It's no problem to there won't be so many species. So that's one thing. But I wanted to comment on the habitats. Yeah, thank you for the comment. It's very we have you here, Padu and Felícia, because you look at the data in a different way than we do. So absolutely, it's true that we should see, we should show how the basic environmental variability across the country. So I think the most important maps are altitude and correlated temperature and precipitation, forest, wetlands, and we have maps for all of these. So it's quite easy to provide people who don't have much idea about geography.
Yes.
I think Milan is mute.
Milan is
Or
here.
he's
Can
frozen.
you see me, hear me?
Now, yes, but it's a bit frozen.
And again frozen.
Yeah.
Mhm. Okay. So I don't know. So
maybe
you have to keep the comment about the habitat types, and I think now we can move to Kuba.
Sorry,
Yeah.
I'm back. Sorry,
Okay.
I don't know what's happening. Lars mentioned that that habitat types could be one of the predictors, and we have maps of diversity of habitat types. Jan Divíšek, I think he is not here, Honza, but he has such data. We use them based on the national habitat mapping. We used them in one paper a couple of years ago. So he has diversity of habitat types in the grid cells. I don't remember now whether they are these grid cells or the larger ones, but for correlation with RAO of traits,
Totally.
it would be, I think, very strong predictor.
Yes.
Please tell Honza to give you the data.
Well, if I can comment on actually both things. So first is the using of spore mass. I'm afraid that the spore is quite different from seed, so there may be criticism that these are not homologous organs. So I'm not sure that this is the best way. And for the habitats, what I have in my mind, and I even did some preliminary analysis with this, was that I took frequencies of species in individual habitats and used such metrics as response and trait predictors. So it was sort of a discriminant analysis in this case, and this showed quite strong patterns and quite interesting ones. Yeah. So and I did this only for herb species because, of course, for including trees would show just that trees grow in forests, which is trivial. So yeah, so habitat type is important, of course.
Yeah, Irenko, maybe.
Just one last remark. It's not only seed mass, it's also the pollen vector and the generative reproduction and some other traits which are connected to this. So maybe it's really easier to do at least one analysis without pteridophytes.
No, but I would say I don't care so much that like seed and spore are like biologically different entities. Like ecologically, they are means of generative dispersal. So we can call it dispersual mass. Of course, then you can argue that dispersules are also some fruits that are heavier, but usually if something is dispersed with fruits, then I don't know.
A short comment. I also won't care. Even in other traits, we are not using something that is fully phylogenetically homologous. For height, it may be attained by leaves or by stems. It's the same thing. And actually, with the spore mass thing, the whole thing may be sorted out by using log transform, which should be used anyway. And if we use either approximate spore mass or lowest value for seed mass, which is essentially orchid seeds, which are almost spore-like, then log transform will sort it out anyway.
Yeah, the seed mass is log transformed, so you're right. I agree with this.
if we are not using zeros.
Sure.
Okay. So, Milan.
So now maybe more like general question how to frame the story around it. So it's an explorative analysis. As Felicia mentioned, we don't have any hypothesis. We have just data, and we are looking for patterns in the data. And when we find patterns, we try to explain them using environmental variables. So that would be such kind of descriptive paper. Like the question is, like what is the pattern of traits in space in a temperate flora, and how is it explained by the environmental variable? Like, do you think it's a good idea to like write a paper like this, or do you have some better ideas?
Well, I think we are so much into this now that I think we should keep going in the idea of the describing. Yeah, I think there are places for such type of papers.
Because I have of course we could make a hypothesis about some traits and test whether these traits are responding to some environmental variables. But after all this experience with the Pladias data, I'm a bit skeptical about it, because if you test such a hypothesis, then you will see that this tested variable, this tested trait, is correlated with some other traits. And now, if you include, you can have some nice hypothesis, you can test it, it will be nicely significant, you will develop a story around it. And then when you include more traits into the data set, you will suddenly see that this trait is correlated with many other traits, and it can be simply like correlation due to something completely different.
Mm-hmm. So, yeah, this is why I thought it was better to work with the strategy axis, because we are not saying this or that trait is important. We are saying, well, okay, this shape or this strategy.
Yeah, maybe Tomáš would like to respond.
Well, I'm just commenting on the issue that Milan raised. And I haven't got much to add, but I think that this takes us back to the trait selection. The trait selection is something that actually determines the set of correlations that we will be getting, and which we will be correlating with the environment. So if we want to framing the exploratory, framing any exploratory analysis essentially depends which traits are included, and we should make clear case we should make clear argument for the thing that the traits are selected according to some kind of a priori knowledge how plants are looking like, and they are not just a random selection of traits that happen to be in the Pladias database.
Yeah, I think with this pre-selection, at least to make more sense and not have duplicates.
Pre-selection would help, but it will have more. It will delete mainly, so to say, strong correlations, but it will not frame the thing. If we say delete clonal traits, as have been discussed here, then we just change the whole framing of the question, which would not make it less meaningful, but it would just be asking a different question. I wonder, this is just a sort of fast idea, whether one should not, no, this is not a very good idea, but I'm going to say, to mention it. Just work with different subsets of traits, defining different frameworks, how one could be looking at the set of traits that our plants are having all together, and to see how these differ in their relationship to the environment. This could be formalized to some way. In spite, I can't now tell how this should be done. But definitely, the basic issue coming to what Milan has said is that the trait selection is something that actually frames the whole thing. this will essentially determine whether the paper will look more exploratory or more hypothesis-based.
Well, yeah, what we could do is try to run the ordination with the traits people usually use. Like if we take leaf traits, height, for example, and put that in the ordination and see how the trends will be, if it's going to be very different from what we have. Because, for example, leaf economic spectrum we have in our second axis. So I don't know if using these selected traits would be so different, because that's the whole point on the ordination, is to try to put all the information together in a way that it makes sense. Even though we are using a lot of traits, at least it should be.
Yeah, anyway, we have to think about it. So,
yeah,
so
yeah.
if you, yeah. So the idea is that if we will use the same 6 traits like Sandra Diaz and run it on quite different scale, so which is Czech Republic, so the expectation will be that the gradient could be similar or not, as she says, she plays, but I'm not sure about how to tackle it now. Jakuba, maybe you want to respond.
Yeah. Maybe we should think about the novelty of the paper, or the prospective paper. And actually, it is also related to what Milan and Tomáš said, that I think that the novelty is that in temperate flora, clonality makes a big difference. Although, well, it was also shown by other papers, written by Tomáš, for instance, but not in this context of the other traits. And also, what is another highlight is the big amount of traits we have. So that is basically something which has no parallels. And concerning the correlations, we should definitely remove these like trivial correlations, like life form and growth form. On the other hand, we have other correlations in the dataset. For instance, height and leaf economic spectrum. That's on the second axis, which is not trivial, because these traits could be independent. Or clonality and hydrophyte life form. So there are many individual correlations or patterns which are not trivial, and are quite interesting. And what is also important, they actually show that, or they very much agree with like common botanical knowledge, and show something which I can see in the field, but now they are displayed like using really nice analysis of large data sets. So this is what I think is really cool about these results.
I still have one sort of related idea that actually comes from the Sandra Diaz paper. What about taking the scores of species in Sandra Diaz paper and just to map our species onto her ordination, to see what part of the overall space that she is having in the analysis is covered by the species that we are having. This would also sort of support the argument that Jakub has made. Let's show what can happen in a flora like this, well, and see how the flora like this looks like.
If I can have a very specific comment on that, sorry. Sorry, am I interrupting you or?
No, I'm essentially finished.
Okay, we just had a new postdoc starting in Budweis in Schussbach's lab, and what he's done is he basically provided a platform for what you just described, where you can go online, and there's a so-called Shiny app, so it's R-based, and you can play around with the Dias paper data.
Wonderful, wonderful.
You can rotate it, and you can add and subtract traits, and then you can load up your own trait matrix and project it into the space of the Dias paper.
Interesting.
Yeah, yeah.
So that would be a very easy, ready-made solution to such a thing.
So it's interesting to do it on several spatial scales because, yeah, then will be some difference to the Sandra scale paper, I guess. So any other comments, maybe Tomáš?
No, sorry, no. I'm just keeping my. It's already aching because it has been raised for such a long time.
So there are a lot of people who are silent. So maybe now it's time for you. No. So if not, thank you, Felícia, once again for this presentation. It was really inspiring.